Welcome to the deep dive. Today, we've got a really clear mission, actually. We're putting together, the ultimate guide for Mr. Martin's grade 8 science class.
That's right.
So if you're working on Criterion B, that's planning and designing your science investigation, this is for you. We want to make all those rules simple and easy to follow.
Exactly. We're shifting gears from just, messing around in the lab to really planning things out so carefully that your results are solid, trustworthy, and, this is key, repeatable. Anyone else should be able to do what you did.
Right, repeatability. That's the goal.
If you can repeat it, you can trust it. And that starts way back in the planning.
Okay, so today we've got 4 main things to cover. First up, planning the experiment itself, figuring out your variables and your hypothesis.
The blueprint, basically.
Yeah, the blueprint. Then second, writing the method. Think of it like your science cookbook.
Step-by-step instructions.
Exactly. Third, getting your data table perfect.
Yeah.
How to organize all those numbers you collect.
Super important for seeing patterns.
And finally, number 4, checking your data quality. We'll talk about precision versus accuracy. Sound good?
Sounds like a plan. Let's jump into that blueprint. Planning the investigation. Okay, so every really good experiment starts with, identifying 3 key things. We call them variables. You absolutely have to know what these are before you even think about starting.
Right. Let's start with the one you actually change, the one you control on purpose. That's the independent variable, right? The IV.
Yeah, spot on. It's the thing you intentionally change to see what effect it has. So, if you're testing how light affects plant growth, the IV is maybe the amount of light, or the hours of light per day. It's the cause.
Okay, the cause. And then there's the effect. That's the dependent variable, the DV.
Precisely. The DV is what you measure to see if changing the IV actually did anything.
Yeah.
In that plant example, the DV would be maybe the height of the plant.
Just height.
Well, you need to be specific, not just height. You need to define how you'll measure it, height measured in centimeters using a ruler, from the soil level to the tip of the tallest leaf. That kind of detail.
Got it. Specificity matters. Okay, so IV is what you change, DV is what you measure. What's the third one?
The controlled variables, the CVs. Now, these can be tricky, but they're maybe the most important for a fair test.
Okay.
These are all the other things, all the other factors that you have to keep exactly the same for every single trial, every setup.
So if I'm testing fertilizer on plants...
Right, then every plant needs the same amount of water, the same type of soil, same size pot, same temperature, same amount of light. Everything except the fertilizer needs to be identical.
Okay, that makes sense. Keep everything else constant.
Yes, but here's where we really need to focus, especially for Mr. Martin's class. It's not quite enough just to list same amount of water. You have to say why it needs to be controlled and, crucially, how you're going to control it.
Hold on. Why the how? If I write down use the same water, isn't that clear?
It sounds clear, yeah, but think about repeatability. If you don't specify how do I know what same amount means? Did you guess? Did you use a leaky cup?
Oh, okay.
But if you write control variable, amount of water, why? Plants need water to grow. Different amounts could affect height. How? We will measure exactly 50 milliliters of tap water using a 100 milliliter graduated cylinder and give it to each plant every day at 8 a.m. See, now that is specific.
Right, okay. That makes it truly repeatable. Anyone can follow those instructions. It removes the guesswork.
Exactly. It ensures a fair test. Without that how, maybe your plant height changed because of water differences, not the fertilizer you were testing. Your results wouldn't mean anything.
Okay, got it. Specify the how. So once we've nailed down the IV, DV, and the CVs with their hows, what's next? The guess.
The educated guess, the hypothesis, and there's a very specific way you need to write this. It's like a formula.
A formula. Okay, lay it on me.
It has to be. If
Yeah.
you describe the change you're making to the IV then, you predict how the DD will change because you have a scientific reason.
If then, because, okay, so for the fertilizer, if we increase the amount of fertilizer given to the plants, then the plants will grow taller because fertilizer provides nutrients essential for plant growth, like that.
Perfect. The because part is crucial. That's where you connect it to actual science, maybe something you learned in class or researched. It shows it's not just a wild guess.
Right, based on some background science. Cool. Anything else for the plan?
Just 2 more quick things for the design. You need a clear list of all your materials and equipment. We call that the apparatus list.
Like beakers, rulers, soil, seeds, everything.
Everything. And ideally, a nice, clear labeled diagram of your experimental setup. how things will look when you actually do the experiment.
So someone could basically build it from your drawing.
That's the idea. Remember, the whole point of this planning stage, criterion B, is making it so clear that someone else could repeat your work exactly.
Okay, blueprint sorted. We know our variables, our hypothesis, our setup. Now we need the instructions, the method. You called it the cookbook.
Yeah, it's literally the recipe for your experiment, a set of super clear, step-by-step instructions. Anyone, even someone who's never done this before, should be able to pick up your method and do exactly what you did.
So structure is important here, I'm guessing.
Absolutely critical. Two main things, numbered steps. 1, 2, 3, 4. And they have to be in chronological order, the actual order you do things.
Right, you can't measure the results before you set up the experiment.
Exactly. And don't assume anything is obvious.
What do you mean?
Well, like if the first step is putting on safety goggles, you write, 1, put on safety goggles. If you need to measure something, you say how you measure it. 2, use the electronic balance to measure 10 grams of salt. Be explicit about every single action.
Even things like stir the mixture.
Yes. Maybe even specify for how long or with what. 3, stir the solution gently with a glass rod for 30 seconds. Think. Could someone misinterpret this? If yes, add more detail.
Okay, total clarity. And connecting back to the variables, you have to say exactly how you change the IV in the steps, right?
Yes, absolutely. Don't just say add different amounts of salt. Your method needs to say specifically. 4, add 5 g of salt to the first beaker. 5, add 10 grams of salt to the second beaker. 6, add 15 grams of salt to the third beaker, and so on. List the exact values you decided on in your plan.
Makes sense. Now, you mentioned repeatability earlier. That comes into the method too, doesn't it? The reliability thing.
Yes, the golden rule. You absolutely must state in your method that you will repeat each measurement.
How many times?
The standard minimum is usually 3 times for each value of your independent variable. So if you're testing 5 different salt concentrations, you run the whole experiment 3 times for the 5G concentration, 3 times for the 10G, and so on.
Okay, 3 times minimum. Why 3? Why not just once if I'm careful?
Great question. Because everyone makes small errors sometimes. Maybe you stop the timer a fraction of a second too late once, or misread a scale slightly. Repeating helps you catch those little inconsistencies, those outliers.
Oh.
Yeah, a result that looks way off compared to the others. If you only do it once, you don't know if it's a genuine result or just a mistake. With 3 trials, you can usually spot the odd one out, and you can calculate an average.
The average. So the repeats make the average more trustworthy.
Exactly. It smooths out those little random errors. So, your method needs a step-like. Repeat steps 4 through 9 3 times for each salt concentration. And also mention how you'll record the results.
Like, record the time taken in seconds in a data table.
Perfect. Just like that. One last tip for the method. Before you start, maybe ask a friend, someone not doing the exact same experiment, to read it.
Like proofreading.
Yeah, like a clarity check. Can they understand it? Did they see any steps missing? Did you forget any safety precautions? It's a really useful final check.
Okay, method written, checked, ready to go. We do the experiment, and now we're getting data. Lots of numbers. How do we handle it?
With a beautifully organized data table. Seriously, don't just scribble numbers down anywhere. Tables are amazing tools. They let you sort things, compare results easily, and spot patterns or trends much faster than looking at a messy list.
Right, structure is key again.
Okay.
Are there rules for tables?
Oh, yes. Very specific rules for good science tables. Rule number 1, and this is a big one. Units go in the column headings only.
Only in the heading. Explain that.
So, if you're measuring temperature, your column heading might be temperature C. The little degree C symbol, the unit, is right there in the heading.
Okay.
Then, in the boxes below that heading, the data cells, you only put the numbers. Like 25.5, 26.0, 24.9. You do not write 25.5 degrees C, 26.0 degrees C in every single box.
Why not? Seems harmless.
It makes the table messy, harder to read quickly. And crucially, imagine trying to calculate an average later if every number has letters after it. You'd have to delete them all. So units only in the header. Numbers, or sometimes words like observations, only in the data cells.
Got it. Header for units, cells for numbers. What else?
Remember those repeats we talked about? Your table structure needs to show them clearly. You'll need separate columns for each trial. Usually trial 1, trial 2, trial 3.
Okay, so I can see all the raw data.
Exactly.
Oh.
And then you need one more column right next to them, average. This is where you'll put the calculated average of your 3 trials for that specific IV value.
Okay, that makes sense. Trial 1, trial 2, trial 3, average. What about how much data we need?
Right. Quantity matters. 2 minimum requirements generally expected. First, You need at least 5 different values for your independent variable.
5, so not just low, medium, high.
Nope, 5 is better. If you're changing temperature, maybe test 10 degrees C, 20 degrees C, 30 degrees C, 40 degrees, and 50 degrees C.
Why 5? Why is 3 not enough?
Because with only 3 points, it's hard to see the real shape of the relationship. Is it a straight line? Does it curve? Does it level off? 5 points gives you a much clearer picture of the trend when you eventually graph it. It makes your conclusion stronger.
Okay, 5 IV levels. And the second requirement.
For each of those 5 IV levels, you need your 3 repeated measurements, minimum.
So 5 IV levels times 3 trials. That's 15 measurements minimum, plus calculating the averages.
You got it. That shows you've been thorough and your results are likely reliable. One last crucial detail for tables. Decimal places.
Ah, yeah. How many numbers after the dot?
Consistency is key. The number of decimal places you write down must match the precision of the instrument you use to measure it.
So if my ruler measures to millimeters, which is 1 decimal place in centimeters, like 12.3 centimeter.
Then all your length measurements in that table must be written to 1 decimal place. Even if you measure something exactly on the line, like 12 centimeters, you must write it as 12.0.
Okay, record to the precision of the tool. What about the average? Does it follow the same rule?
Yes, this is important. Your calculated average should be rounded to the same number of decimal places as your raw measurements. Keep that consistency throughout the table.
Got it. Consistent decimal places matching the equipment. Any tips for making the table look neat?
Yeah, definitely use a ruler if you're drawing it by hand. Clear lines make a huge difference. If you're using a computer, use the table tools, put borders on, maybe center the numbers, and crucially, record your results directly into the table as you get them. Don't try to remember them later.
Good point. Write it down immediately.
Okay, so you've run the experiment, you've filled your beautiful table with data, showing repeats and averages. Now, how do you know if that data is actually any good?
Right. Is it trustworthy? This is where precision and accuracy come in, isn't it? They sound similar, but they're different.
Very different, yeah. It's a common point of confusion.
Good.
Let's use the classic dartboard analogy.
Hey, I like analogies.
Imagine you throw 3 darts. Precision is about how close your darts are to each other. If all 3 darts land really close together, maybe in the top left corner, your throws were precise. You were consistent.
Okay, clustered together,
Yeah.
precise, even if it's not near the middle.
Exactly. That's where accuracy comes in.
Yeah.
Accuracy is about how close your darts are to the actual target, the bullseye.
So if my darts are all clustered in the corner, they're precise, but not accurate.
Spot on. And if your darts are kind of scattered all around the bullseye, maybe one in it, one above, one below, they might average out to be accurate, but the throws themselves weren't very precise.
Ah, okay. So you can be precise, but inaccurate, grouped, but wrong spot, or accurate, but imprecise, scattered around the right spot.
You got it. And ideally, in science, what do we want?
Both. We want our results to be clustered together and centered on the right answer, precise and accurate.
That's the goal. If your results are precise but inaccurate, it often points to a problem with your equipment or setup, something we call a systematic error.
Okay, you mentioned uncertainty earlier, too. Is this related, systematic something?
Yes, exactly. There are 2 main types of uncertainty or error we think about. First is systematic uncertainty or systematic error. This usually comes from your equipment or your setup. Like maybe the weighing scale wasn't zeroed properly, so every measurement is off by, say, 0.2 grams. Or the thermometer always reads 1 degree too high. It's a consistent error affecting all measurements in the same way.
So that would make my results inaccurate, even if I measure very carefully each time.
Precisely. It leads to precise but inaccurate results. The error is built into the system.
Okay, what's the other type?
The other is random uncertainty or random error. This comes from, well, randomness, mostly human factors.
Like my reaction time using a stopwatch.
Perfect example. Your reaction time might be slightly different each time you press start or stop. Or maybe you read the volume in a measuring cylinder slightly differently each time because of the meniscus, that curved water line. These are small, unpredictable variations.
So random errors affect precision. They make the results more scattered.
Generally, yes. They make your repeated measurements less close to each other. Now, think back to our repeats, doing the experiment 3 times. Which type of error does repeating help with?
The random kind. Because if I time it 3 times, my slightly too fast and slightly too slow reaction times might average out.
Exactly. Repetition and averaging are fantastic for reducing the impact of random errors. But and this is critical.
Oh.
Repeating the experiment does absolutely nothing to fix a systematic error. If your scale is always reading zero point 0.2 grams too high, averaging 3 measurements from that scale will still give you an average that's zero point 0.2 grams too high.
Right. The system's error is still there.
Which is why getting section 1 right, carefully checking your equipment, controlling those variables properly is so important. You need to minimize systematic errors from the start. Repeating only helps with the random stuff. #outro.
Wow. Okay. That brings it all together. We've covered a lot, but it feels like a clear path now.
Yeah.
Let's quickly recap those 4 steps for everyone listening, especially Mr. Martin's class.
Good idea.
Okay, step 1. Plan your variables. Know your IV, your DV, and list your CVs, explaining how you'll control them.
Don't forget the how.
Step 2. Write that hypothesis using the magic formula. If IV change, then DV prediction, because science reason.
Back it up with science.
Step 3. Write your method. Numbered steps, chronological order, super detailed, and make sure you include repeating each measurement at least 3 times.
Reliability is key.
And step 4, create that perfect data table. Units only in the header, columns for trials, and the average, at least 5 IV values, and keep those decimal places consistent.
Nailed it.
So, what's the big takeaway here, if you could boil it down?
I think the main thing is this. Careful, clear planning, doing criterion B really well is what makes your results trustworthy. It's the difference between just doing an experiment and doing good science.
Good science being science that other people can trust and repeat.
Exactly. That's how science moves forward. People building on reliable work done by others. Your clear plan is the foundation for that.
Okay, great. So we always like to leave you with something to think about. A little provocative thought.
Let's hear it.
Think about a simple experiment like dropping a ball from a certain height and timing how long it takes to hit the ground. Which type of error do you think is usually harder to completely get rid of? Is it the potential error in measuring the exact drop height with a ruler? Maybe the ruler itself is slightly off, or you don't hold it perfectly straight each time. That sounds like it could be systematic.
Could be.
Or is it the slight variations in reaction time when starting and stopping the stopwatch, especially if maybe 3 different students are timing it? That sounds more like random error.
Yeah.
So, which is tough for it to eliminate completely in a typical classroom setting.
Yep.
The potential systematic error in the measurement tool or the random error from the human timers.
That's a good one. Thinking about equipment limitations versus human variability. Definitely something to mull over as you plan your own investigations.
Absolutely. Food for thought. Well, that's all the time we have for this deep dive.
Hope that was helpful.
We'll catch you on the next one.